Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled An Optimized AAV Sample Preparation Method for Robust Quantification of Viral Titers by PCR. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Technova, who developed the content for this presentation. Technova makes solutions possible. They help accelerate breakthroughs for customers in cell and gene therapy, bioproduction, molecular diagnostics, and synthetic biology by providing faster access to high-quality, fully customizable agar plates, microbial culture media, buffers, and reagents. Their proprietary modular manufacturing platform enables them to deliver consistent customer products quickly, making it seamless for customers to scale from RUO to GMP as they work to develop novel therapies that can help people live longer, healthier lives. I'd now like to introduce our speaker for today's event. Zoe Bowman is a research associate at Technova, where she focuses on advanced analytical development and tissue culture work for their novel AAV product portfolio. She is a key team member responsible for the launch of the company's first proprietary product, the AAV Tech AEX Buffer Screening Kit, and is working on solutions to help common bioprocessing bottlenecks, such as AAV sample treatment prior to analytical testing. But now, without further ado, let's go ahead and hand things over to our speaker for today. Zoe Bowman, you may begin when you're ready.
Okay, so thank you so much for the introduction, and thank you to everyone for joining us in this talk today. I am so excited to be talking about all the work you're doing at Technova, and specifically around our AAV sample preparation for PCR. Next slide, please. Given the wide range of applications for PCR, I'm really curious to know what serotypes everyone is working with.
So appearing on everyone's screen right now is a polling question, and you can participate by selecting as many of the options that you see in front of you that applies to you, and then click submit. And as you heard, the question that we're wondering is, what serotypes do you work with? Your answer options are 2, 6, 8, 9, or engineered capsid. Give everyone a chance to consider their answer. Again, you can select more than one, and then click submit to get your answer in. All right, so we have our submissions here. Let's look at our polling breakdown from the audience. All right, we have 60% of you selecting engineered capsid, and then 80% for 9, 20% of you selecting 8, 40% selecting 6, and then 20% of you selecting 2 of them. So thank you so much for your participation, and it's back to you, Zoe.
Awesome, thank you so much for answering that poll question. It's great to see that everyone's working with different serotypes out there. Here at Technova, we are working with multiple serotypes ourselves in order to make sure that we meet the needs of everyone in our field. All right, so for those of you who aren't familiar with Technova, I'd like to do a quick introduction into our company before really jumping into the bulk of this talk. Here at Technova, we make solutions possible. We make a wide range of products, including pre-poured media plates, cell culture media and supplements, as well as molecular biology reagents. And these products have a wide range of applications spanning from CDMOs, academia, diagnostics, and of course the bioprocessing field where the cell and gene therapy space lives. And in an industry where a lot of companies are making buffers and reagents, what makes Technova so special? So here at Technova, we are able to deliver our high-quality custom products and reagents with short turnaround times due to the fact that we use something called modular manufacturing. So modular manufacturing means that we can seamlessly swap out different factors in our processes, such as testing criteria, packaging configurations, etc., in order to meet everyone's needs. All right, so now that I've said a little bit about what we make at Technova and how we make it, I'd like to go and do a brief history on our company and also share our plans for the future. Thank you, Ryan. So Technova was established in 1996. We've been around for quite a while, and we started by making buffers and reagents that were most commonly found in many biologic labs. Then in 2017, our company started to invest heavily into our research and development department. This was a very important move for Technova as it has brought us into the cell and gene therapy space and to those customers that we really want to work with. Because of our processes using modular manufacturing, we believe that we really are a perfect fit for this field. We love the cell and gene customers that we work with currently, and we are really excited to work with more in the future. And then after a few years of investing, we were actually able to open up our own research and development lab where we could begin our work with AAV. Next slide, please. Thank you. Okay, so as many of you are probably very familiar with the AAV workflow, shown here is our in-house platform at Technova, where we do everything end-to-end ourselves, starting with our plasmid production, where we clone and purify our plasmids, and then moving on to our expansion, where we will grow our cells in shake flasks and bioreactors. And then after harvesting our virus, we then move on to our purification, where we do our filtering. We do have a TFF set up in our lab, as well as 2 ACTAs for any chromatography needs that we have. And then, of course, throughout this entire production and purification, we are doing a suite of analytics. Because we are producing and purifying AAV all the time in our lab, it has given us the insight into being able to address the pain points of the whole workflow. The major pain point that I'd like to be talking to you guys about today is, of course, the unmet need for standardized analytical measurements, and more specifically, the unmet need for standardized and established PCR protocols. We all know that in order to feel confident about your experiments and your processes, analytics really are key, which is why we decided that it was so important for us to dedicate our time to establishing a standardized protocol. Next slide, please. So before we jump into our protocol, I'd like to first talk about the overarching analytics for the AAV field. Starting with our capsid quantification, many of us know capsids are made up of viral proteins that, of course, encapsulate our viral genome. This measurement is very important as it tells you how many AAV particles you have in your sample. And while it may not give you a hint as to how many full, partial, or empty capsids you have, it's still a very important measurement when trying to understand what's going on with your AAV. There are a few different ways that you can do this measurement, such as Western blots, Stunners. The most common that we found in our field through literature and talking to cell and gene therapy experts is the ELISA assay, and specifically the Progen ELISA kits. And while we know that these kits are great for wild types, of course we understand that those of you who are working with your synthetic capsids, these kits are not commercially available and lead you to creating your own assays. The next analytic I'd like to talk about is our genome quantification. This is a very important quantification as it tells you exactly how many transgenes you have in your sample, which is ultimately how you are going to dose your AAV drug. This quantification can be done a few different ways. Most commonly in our field is PCR, so you can do qPCR, digital droplet PCR, or digital PCR. And typically when you are doing PCR, there is some sort of sample preparation involved and also making sure that you're getting into your range of detection. And as we have found through scouring literature, the sample preparation is actually not standardized. And not only is the sample preparation not standardized, but the PCR protocol itself isn't, as it does require target-specific designs. And then moving on to our last analytic I'd like to go over is, of course, our empty-full ratio. It is one of the most challenging, yet one of the most crucial, as it does tell you your percentage of full capsids that you have. It is very important when you are getting into your final drug product. There are a few different ways that you can do this empty-full measurement. The most common that we found is a PCR over ELISA measurement, but there are other ways to do it as well, such as AUC or CDMS. And while these other orthogonal methods do exist, they're not always easily accessible and or established. So while we do a wide suite of analytics here at Technova, we are primarily using our Progen ELISA assays for the capsid quantification. We are using digital PCR for the genome quantification, and we are using a combination of PCR over ELISA with our orthogonal methods for our empty-full ratio. So while we were reflecting on our analytics, what actually sparked this project is we were looking into our empty-full ratio. We were looking at our PCR over ELISA measurement versus our other orthogonal methods, and the titers were not aligning. So we wanted to make sure that we could get those methods to align. We know that because we're using the Progen ELISA kits, that assay is already established and standardized, and our orthogonal methods are also already established. So we knew we could dig further into our PCR sample preparation and the measurements and see if we could get those measurements to align. Next slide, please. Okay, so to begin our journey in investing our PCR sample preparation, we wanted to outline some of the challenges that we wanted to face. So starting with our first challenge is the DNase treatment itself. While many of us are doing a nuclease treatment when we are harvesting our virus, it is still very important that we have a DNase treatment pre-PCR as well, as it will remove all of that exogenous DNA that is outside of our capsids. And removing that exogenous DNA is very important when you are trying to get an accurate quantification. The second challenge I'd like to talk about is a concentration of EDTA that could potentially be too low. We knew that we wanted some EDTA in our sample prep, as it does prevent the degradation of DNA and will also aid in the DNase inactivation. And then moving on to our third challenge, which is very similar to the second, except we wanted to address a concentration of EDTA that could potentially be too high. So while EDTA does do a lot of good for our samples, it is also a well-known PCR inhibitor. So balancing that concentration of EDTA was really important. And our fourth challenge we wanted to look into was the need for a DNA extraction kit. We know that viral DNA is commonly extracted using spin column extraction kits, which these kits can be very costly and timely. And while we know that these kits are crucial for many other viruses, we wanted to investigate further to determine if it was also necessary for AAV. And of course, being a buffer company, we know that the impact that buffers have on biologics is great, so we wanted to look into a dilution buffer. We wanted to create a buffer that could stabilize our AAV, prevent an aggregation, and aid in our capsid disruption. Which leads us to our last challenge, which of course is that capsid disruption. In order to effectively get an accurate quantification, you need to remove all of your capsids so that that viral DNA that's inside can be exposed for your PCR reaction. All right, next slide, please. So now that we've looked at our challenges, let's go ahead and talk about how we have addressed them all. So the first set of experiments was done in a DOE style, where we were able to address the first 4 challenges. With this DOE, we tested 4 different time points for our DNase activation. We tested 4 different concentrations for our EDTA. We tested the need for a DNase inactivation method by heat or by dilution. We also tested if we needed an extraction. So while you can see here, we do have these response contour plots that we got after completing the 20 experiments, gathering our data, plugging it back into our DOE. As you can see with these response contour plots, they're a little bit overwhelming at first glance, but they did provide us excellent data. As you can see on the right-hand side of the graph, we have a titers ranging from 3.3 E10, which is shown in that light blue color, all the way to 3.4 E11, which is shown in that dark purple color. We were very surprised to see such a wide range of titers here, but as you can see on our response contour plots, there is a clear area where that dark purple has taken over, where our optimal condition lies. Next slide, please. So here we have our optimal condition highlighted in orange over here. So our optimal condition that our DOE gave us was a DNase activation time of 30 minutes, an EDTA concentration of 25 millimolar, DNase inactivation by dilution, and we found that we did not need an extraction kit. This DOE was very helpful in addressing those first 4 challenges, and it has given us a protocol that is less time consuming and less costly with the removal of the extraction kit. Okay, and before we actually get on to the last 2 challenges, I'd like to do another poll question.
Yes, so this should be appearing on everyone's screen right now. As with the previous question, you can select more than one option before clicking submit. The question that we're asking you is, what do you dilute your AAV sample in prior to PCR analysis? Your answer options are water, TE, commercial PCR buffer, buffer plus surfactant, or other. Give everyone some time to consider an answer, selecting more than one, and then to the question again of what do you dilute your AAV sample in prior to PCR analysis. Looks like most of you have your vote in, so thank you so much for your participation on this. Let's look at your results here. We have 29% of you selecting water, 14% for TE, 0% of you selecting commercial PCR buffer, but then 43% for buffer and surfactant, and then 29% for other. So thank you again, and again, back to you, Zoe.
Awesome. Thank you so much, everyone, for answering that poll question. Speaking of dilution buffers, let's go ahead and talk about our fifth challenge, which is, of course, our dilution buffer. So to begin this, we scoured literature to find the most common buffers that were being used. As you can see on this graph to the left with our dilution buffers, we have plotted the most common that we found. Common buffers including water, TE, PBS, and then a combination of these buffers with a surfactant as well. As you can see here, we have, for example, TE plus 5% tween 20 plotted. And then as we were screening buffers and playing around with concentrations of everything, we were actually able to find a Technova dilution buffer that gave us an enrichment of 44% compared to our water. And while this buffer will be proprietary, I can share that it does have properties aiding in AAV stability, aids in capsid lysis, and also prevents aggregation. So moving on to our last challenge, which is our capsid disruption. As you can see here on this graph to the right, we have 3 different serotypes plotted. This graph shows our heat-treated versus our not heat-treated, so our capsid disrupted versus our not capsid disruption. So in order to get this graph, what we did is we took samples from each of these serotypes, we DNA treated them, we diluted them in our Technova dilution buffer, and then we actually split the samples, and half of them received an additional heat treatment pre-PCR of 95C for 10 minutes in order to capsid disrupt, and then the other half of our samples received no additional heat treatment. So as you can see here with our graph, we show for AAV2 above a 30% increase, and for AAV6 and AAV8, we show above a 20% increase. So as you can see, this additional heat treatment before PCR is very necessary in order to get that viral DNA out and allow it for quantification of our PCR. Okay, so now that we've gone over all of the challenges, we wanted to, of course, do a robustness study. Next slide, please. Thank you. So we wanted to do this robustness study because we know not everyone is working with digital PCR like we are. We know not everyone is working with the same serotypes, and we also know that not everyone is working with the same purification steps. So with this study, we wanted to cover everything here. Really quickly, just looking at the differences between the methods. So our method 1 had a spin column extraction kit. Our method 2 is our DOE output. And then our method 3 is our final protocol, which is our DOE output, plus an additional Technova dilution buffer and that capsid disruption step. So looking at the graphs, you can see we have 2 graphs plotted for AAV2. These results were also confirmed for AAV6 and AAV8 as well. On the left-hand side, we have our qPCR graph. On the right-hand side, we have our digital PCR graph. And if you look at the x-axis, we do have steps throughout the purification, going from harvest lysate to our filtrate, our affinity chromatography load, our affinity chromatography eluate, and our AEX peak. And just a visual, if you want to look at these 2 graphs compared to each other, you can see that they do follow the same trends, and they do have similar titers. Of course, your qPCR is going to overshoot your titer just a little bit because it is using that standard curve. It's not quite as precise as our digital PCR is, but they do follow the same trends, and they do have very similar titers. So with this study concluded, we do feel like we have a very robust protocol. And speaking of our protocol, I'd like to go ahead and walk you through our protocol as well as sharing a little bit of an announcement on our next slide. So with our work wrapping up for our AAV sample preparation for PCR, I'm very excited to announce that we will be launching a kit by the end of this year that will include our final protocol, as well as all of the reagents that you will need to follow it. Like I said, it is going to be launched at the end of the year, but we will be offering sample kits before then, so please feel free to reach out if you would like to receive a sample kit and try out our protocol. Okay, so let's go and walk you guys through the protocol really quick. So starting with our DNase treatment, we will be providing our DNase as well as a modified 10x DNase reaction buffer. Once these reagents are combined with your AAV sample, you will then heat treat for 30 minutes at 37°C. This is going to remove all of that exogenous DNA. And then moving on to a dilution, something that is really cool and unique about this kit is that we will be giving you guys the option of how you'd like to receive your dilution buffer. So the first way is you will have a 10x dilution buffer, which you will then dilute to 1x, and then you can use however you'd like. But we do know that a lot of us are doing serial dilutions, which is why we wanted to give the option of a pre-filled dilution plate. So these plates will be 96 well PCR plates filled with 90 microliters each of our Technova dilution buffer. So if you choose to get these plates, you will get them, you will just spin them down, and then you can just do your dilutions. They're all ready to go for you. Then after your dilutions are completed, you will then move on to a capsid disruption step, which you will place your samples into a thermocycler and heat for 10 minutes at 95°C. And then once your capsids are disrupted and your viral genome is released, you will be ready to do any PCR that you would like to do. And I'd like to go and highlight some key advantages here. So this kit, it will allow you to save time and cost, especially if you get our dilution plates. You'll be saving that time that you would be taking to aliquot your dilution buffer. And of course, from our first method to our third method, that removal of the extraction kit saving us time and cost as well. This kit is designed to be very easy to use. As you can see, it's just 4 short steps. And of course, we have the robustness with our last slide. And while these advantages are great, the key advantage that I really want to highlight is the accuracy of your titers that you will get with this kit. We've done a lot of work to prove that these titers we are getting are accurate and reliable and reproducible. We have done this protocol with a standard and received results that were very similar to the titer that was reported to us. And then, of course, looping back to what sparked this project, looking at our empty-full ratios. So with this protocol, we were able to look at our PCR over ELISA versus our orthogonal methods, and our titers do align very well. So we are very confident in this kit. Next slide, please. And with all that being said, I'd like to give a big thank you to my team and a special thank you to Eric Lustre, as he was the other lead scientist on this project. We have a great research and development department here with incredible scientists, and without them, our AAV novel products would not be possible. And that is the end of my talk. Thank you all so much for being here and listening.
Well, thank you very much for that insightful presentation. As we move into our Q&A session, I'd like to direct the audience's attention towards the handouts module, where you'll find some additional documentation related to today's presentation to download for some further reading. I'd also like to invite the audience to keep sending their questions or comments right now using the questions window for this Q&A portion of the webinar. Now, I've already received some questions for you, so we'll get ourselves started with those. The very first question is wondering, will the PCR test kit work for ddPCR?
So while we have not tested with digital droplet PCRs, we do not have a platform here at Technova. We understand that the technology between digital droplet PCR and digital PCR are very similar, so we do believe that this kit will work with digital droplet PCR as well.
Okay, very good, thank you. Next question here is wondering, have you tested with a standard?
Yes, we have tested with a standard. We have tested with an ATCC standard and reported results that were very similar to the titer that they had reported.
Very good, thank you. The next question here is wondering, how do you know that your protocol is accurate?
So we know that our protocol is accurate because we have, because with those challenges that we have outlined and we've accomplished them all, we know that with the DNA treatment, we are removing all of our exogenous DNA. We know that with our EDTA concentration being balanced, we know that we are protecting our viral DNA, as well as it is helping with our DNA inactivation. And of course, with our Technova dilution buffer, with its properties that aid in AAV stability, prevent aggregation, and aid in that capsid destruction step. And also, because we have that capsid disruption step, we are able to release all of that viral DNA. With all of these challenges being faced, we know that we have an accurate titer and protocol.
Perfect. All right. The next question here is curious, what primer and probe do you use?
So for the data that I have shown here today, we have been using an ITR primer and probe, but we are always doing a gene of interest primer and probe as well. So yeah, and in the future, we will be working on doing more primer and probes that are steering away from the ITRs and moving towards something on the AAV backbone, as well as our gene of interest.
Okay, very good. Thank you so much for that answer and for all of your answers today. However, we have reached the end of our Q&A portion for the webinar. If we couldn't attend to your questions, know that the team at Alpha Technova will follow up with you, or if you have some further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, there's a link to view the recording of this event, which I will include in the chat box here for you, and you can share this link with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speaker for her wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.